Right. Okay, good afternoon and welcome everybody to this Statistical Mechanics in Chemistry and Biology SMCB seminar series. We had been running this seminar series for a while now. Today is going to be the 19th seminar in this series. And the generally the purpose or motivation behind having this series was to introduce young students, both including undergraduate and graduate students, to introduce them to modern applications of statistical mechanics to various applications in chemistry and biology and so on. So we take this, we request all the speakers to spend extended amount of time on introducing the concepts and ideas, particularly for the young researchers, and hopefully they'll be motivated to pursue this line of research. So it's a pleasure to have today Dr. Arnab Bhattacherjee as today's speaker. So I'll just give a kind of a brief introduction about Arnab. So Dr. Arnab Bhattacherjee is currently an associate professor in the School of Computational and Integrative Sciences in Jawaharlal Nehru University, Delhi. So he did his PhD from Delhi University in 2010, and he had a couple of postdoctoral research stints at Lund University, Sweden, and then Weizmann University. And he came back to India in 2015 and joined JNU. And his broad area of research is understanding genome biophysics and the structure-function relation, and also disease and medical conditions related to these problems. So in general, he uses various computational biology technique and theoretical biophysics principles. He has received several recognition and awards. A few of them are, he has been elected the Associate of Indian Academy of Sciences. He's recipient of INSA Medal for Young Scientist in 2022. He also got CRSI Young Scientist Award and also Humboldt Fellowship for Experienced Researchers from the Humboldt Foundation, Germany. So today he is going to talk to us about nuclear traffic and transport, a rule of faster protein communication on DNA. So with that, I request Dr. Bhattacherjee to start with his presentation. Or no, please.
Yeah, thank you, Shumanda. I'll start sharing my screen.
Yes.
Okay. So it is visible, right?
Yes, it is visible.
Okay, great. Okay. So I'll speak on nuclear traffic and transport rules of faster protein communications on DNA. And before I start, I would like to thank the organizer, especially Shumanda, who contacted me for this opportunity to present our work here. So our lab is mainly interested in protein DNA interactions and the biophysics behind these kind of interactions. So I'll start with giving a very basic description of the process and why it is important. So here on the left hand side, these are the different processes which are extremely critical for our cellular systems. For example, transcription, DNA replication, DNA repair, and transcriptional regulations. So all these processes, one common step in all these processes is that a protein, a specific protein, needs to bind at a specific site on DNA. So if you see a cell nucleus, you have a huge amount of DNA base pairs, out of which this protein, let's say, this binds specifically with a certain patch of the DNA. So the basic question that we are interested in is that how do proteins search their specific sites on genomic DNA, which is large, which is comprises of millions of or billions of base pairs, and out of that, how it finds its specific site. That is a major question that we are interested in. So a bit of history we'll discuss here. It was in 1970s, there are two back-to-back JMB papers by Riggs, who worked on lac repressor protein. So this is the DNA, and this is RNA polymerase. So RNA polymerase comes, bind. This is the operator region, and it binds and it represses the corresponding protein. Now, there is a repressor protein. If it binds here, then RNA polymerase cannot go and it cannot express the protein. So this it works like a obstacle towards the expression of this protein. Now, the important part is that in probably third back to back, the second paper of the JMB probably, they have shown that this binding or the association kinetics of this lac repressor protein is very high, in the order of 10 to the power 9 mole inverse second inverse. And they argued that this rate is roughly 100-fold, 100 times faster compared to the upper limit estimated for an ordinary diffusion controlled reactions. So what was the explanation of this faster kinetics? And it took around 10 more years to give an explanation. And they said that there is a facilitated diffusion mechanism operates. So what is that? They said that it is not, so let's say this is the DNA and this is the target site. These are the, so it's not like that only the protein comes, diffuses and reaches on the DNA, but it performs several other motion. For example, we have shown here the diffusion. The protein can move along the DNA also, and therefore it is kind of one-dimensional movement instead of a three-dimensional movement. The protein can also hop on the DNA. So this is basically small jumps, which kind of speed up its search process. And also, if two segments of DNA are nearby, the protein can move from one segment to another segment. This is known as intersegmental transfer. So the idea of facilitated diffusion is that it's not the only 3D diffusion through which the protein finds its target site on the DNA. Rather, it's a combination of all these processes: 3D diffusion, intersegmental transfer, sliding, and hopping, using which it finds its target site much more efficiently compared to a 3D diffusion reaction. However, okay, so if this is the picture, now, if one knows the rates of these individual movements, then one can find out the target search time. So, for example, if you take this kind of picture where you can the protein can from solution can come on the DNA with a rate k on, and it can dissociates off the DNA with a rate k off, and it can move on the DNA with a rate, let's say u, which is one-dimensional movement on the DNA. Then one can write a set of master equations where, let's say, this Fn is n is the position of the protein on the DNA. So the time evolution of this state Fn will change. So from n state, it can go to, with the sliding or the movement on the DNA, it can go to neighboring states n+1 and n-1, as because this is stochastic movement. Or it can, with a k of rate, it can dissociate off to the solution phase. So similarly, if the these two end positions, 1 and lth position, so from one position, it cannot do any more one-dimensional movement on this side. Rather, only one side is available. So it can move to u with a u rate. It can move to f2 side and not on the there is no more side beyond 1. So similarly, at the lth side, these are the two boundary conditions. And from L site, it can go to L-1, and there are no more sites beyond L. So, and the solution phase we can write like this: that from solution, it can come with a rate k on to any site on the DNA. Now, if these are the equations, then one can do simple Laplace transformation of these equations to get an simple algebraic forms. And from that, one can find out the target search time using this kind of operation. So what we have done is this can be shown that the target search time changes with parameter lambda in this fashion. So lambda is given by u by k of. Okay. Now u, this is one-dimensional movement along the DNA. So that means this is 1D movement, and k of is kind of more k of. That means the protein will perform more 3D diffusion. So this is 3D corresponding 3D rates. So this lambda kind of gives you that how long the protein can move along the DNA without dissociating from it. So we call this as scanning length. So now you can see if U is very high compared to K of, that means the protein will predominantly do one-dimensional movement along the DNA. Whereas if U is much smaller than K of, the protein will predominantly perform 3D diffusion and will be on DNA for very less time. So here, if you see that lambda less than equal 1, so the time is high. For lambda very greater than very greater than 1, lambda is very greater than the length of the DNA, which in this case is around 200 base pairs or so. So then again the time increases, but the time is minimum when there is a competition between these two or the combination bit of these two. So which suggests again the facility diffusion concept that to get the fastest target search time, you need a combination of 3D and 1D search processes, search dynamics. Okay, while this process is this analytical approach is simple and very efficient to find out this target search time, given that this experimental kinetic rates are available, the obvious and major problem is that you do not have any structural information, right? However, in many cases, this structural information could be very important. So, for example, in 2000, just a second, I cannot see this. Okay, Barg later on published one paper in Science, which where they suggested that this same lac repressor protein, which they argued initially that it performs facility diffusion, they showed that this similar facility diffusion can be observed in living cell as well. So this was the first experiment done in living cell, and they have shown that again you can. They reported this facility diffusion, but here they have noticed something more interesting. They said that the repressor protein, it frequently slides over the site where it needs to bind. So more than 90% times it slide over it, but it skipped that slide. It couldn't recognize the site properly, and after many attempts, it could find the site. So that means there could be problems that if you are close to the target site, still the protein may not be able to recognize the site. So structural informations in this case is important to understand why that happens. Another very interesting study was done by Professor Biman Bagchi and Sanizi's group, is that they were trying to measure that during this one-dimensional movement, which we call sliding. So let's say this is the protein and this is the DNA, and the protein moves along the DNA. So if it is moving along the DNA, one-dimensional movement, so you can measure the diffusivity or diffusion constant, and you just plot it with 1/r. And they found for many proteins, the correlation is quite poor, 0.52. So an elegant equation was proposed that this is not the case that the protein only moves along this way. Rather, they have hypothesized that when the protein moves, it also rotates along the DNA. And then a new equation was proposed by given by this equation, and it was shown that if now this one-dimensional diffusion coefficient is plotted against this parameter, you get a fantastic correlation of around 94%. So we took up this and we developed a model, coarse-grained model, because obviously this this process, the searching process is quite time consuming, and so far atomistic simulation couldn't achieve this very efficiently. So we have developed a simple coarse-grained model where the protein is presented through C alpha representation, and the DNA is 3 sites per nucleotide. So 1 phosphate, 1 sugar, and 1 corresponding base. and a detailed interactions. We have base stacking, cross stacking, base pairing. They are a little bit complicated. Nevertheless, without going into much details into those energy functions, what we found is that if you have a DNA and you have a protein, and this protein, the green portion is a DNA binding patch. So when I say DNA binding patch, most likely that this patch has a high concentration of positive charges, as because DNA is negatively charged polymer. So this electrostatic interaction plays a major role in protein DNA interactions. So we simulated this system, and what we found is that what previous study said, it's actually the protein moves along the DNA. So it moves along the DNA, and it also simultaneously it rotates along the major groove of the DNA. So this is called rotation coupled sliding movement of the protein. So this was quite nice, and it keeps on doing that. So there are several other movements that we could capture, and there are studies in support of that. For example, if you have two DNA nearby, two segment of DNA, and you can see that if there is a protein which has two binding patches, these are the patches, red one and this cyan color. So if you have protein with, let's say, a disordered tail, where the tail has some positive charges, or two domains where both the domains contain DNA patches, DNA binding patches, then this protein okay, yeah. So then this protein can move on the DNA, and whenever it feels the adjacent DNA segment nearby, it can move from the first segment to the second segment by a transient, by forming a, this is the transient bridge complex, and then it moved to the other segment. So this has been studied extensively, that how the free energy surface looks like, and this is the corresponding the bridging complex, and then it moves between these two segments. And so this, the purpose of doing this is that this protein can efficiently scan more part of the DNA. So this is kind of a way to speed up its search process. Okay, so coming to the nuclear system, if you see, this search process is very similar to that what we experience in real life. So for example, if you would like to move from one place to another place, for this case, I have shown here the New Delhi railway station to JNU. And if you see it on Google Map, you will get a plot like this. And there are several things. So for example, you see these colors, red and these colors, that means there are traffics. So similarly, inside nucleus, there are many other copies of proteins, right? So these proteins act like crowders. So this protein, for example, would like to move on the DNA and find the target site, how it will do, because there are many more crowder molecules or many more protein molecules. So how do they affect its search process? That is one question that we have extensively looked into. Second aspect could be that sometimes you can see there is another route available. So this route was not suggested by the Google, probably because this route has probably it's a longer one, or it could also happen that the this route has a road closure, right? So similarly on DNA inside cell nucleus, you have DNA, and many sites could be blocked by several other proteins which have already occupied their specific sites, or could be some other reason, but the roadblocks are possible. And then one protein, how it will move bypassing these roadblocks, is interesting phenomena to investigate. Another important phenomena is that let's say this is our target site, and these are the sites. After all, this target site is nothing but a combination of AT and GC, these four base pairs. So these are the other target sites which have very high sequence similarity with these target sites. We call them as pseudo sites. So this is like, this potholes on the road. So when you have potholes, you have to drive slowly, otherwise you will get stuck into this pothole. On the other hand, if you move very fast, then you can bypass these potholes because you are moving very fast. Your car will not, get trapped here. But then there is a possibility that you may not be able to stop exactly at your correct destination. So the same feature happens here, that the protein, if it is moving very fast on the DNA, then it may bypass these pseudosites, but then it may also possible that it may not recognize this target site. It can move past through this site. On the other way, if it is moving very slowly, then it will definitely able to recognize this site. But on the other hand, it can get also trapped into these pseudo target sites. So there it needs the between this slow and fast dynamics, and protein usually does this. So we are trying to understand here that how protein balances all these different movement and efficiently search their target sites on DNA. So this is Crowder study. I'll just show a brief result here. Is that we try to understand how protein moves through the crowded environment. One of the major finding that we got is that when there are crowders, and this is the DNA, there is a depletion layer forms, and the origin of this depletion layer is completely entropic. So this depletion layer is a region where no crowders can enter into. So when this protein enters within this depletion region, then the protein kind of doesn't feel this crowder, because the microviscosity within this depletion region is much lower compared to macroviscosity in the bulk because of the crowder molecules. A similar observation has recently been supported by experimental work from Shobhan Sen's group from our university only. So experimentally, it was also suggested that this molecular crowding enhances the facilitated diffusion of two human glycolysis proteins. So what they have done, they have taken a protein and they have taken a DNA, and they have two sites, and they were trying to measure the transfer probability between these two sites by the protein. So they have seen when they have a crowder PEG 8K, they see high transfer rate. So that means that this crowding actually facilitates this kind of transfer from one side to another side. That means the protein is moving efficiently over the DNA in presence of crowders. So what we found is that one feature is obviously this depletion. Because of this depletion zone, the protein can move efficiently, as because it is experiencing the microviscosity once it is inside the depletion zone. Second is, if the protein interacts slightly with the crowders, then its diffusivity is even higher, because this crowder in that case can carry this protein through along the DNA. But then the protein, as because it is slightly off from the DNA, it doesn't feel the friction or the it doesn't feel the energetic barrier that it needs when it moves very close to the DNA. So basically, we have found that if you compare this diffusivity, then it increases for a moderate Crowder protein interaction strength, and it goes down in both the sides, suggesting that this protein Crowder interaction can actually facilitate the search process even more. So these are some of the papers that we have published in this area. And this is the kind of movement that the protein. So here the protein is moving mainly on the DNA, as because the protein doesn't feel any. So these are inert crowders, so the protein doesn't feel any attraction from the crowders. But here the protein is, you can see, is mostly carried away by these crowder molecules, and it is slightly off the DNA. But although it can feel the DNA, but it is moving much smoother. The the landscape is much smoother here. The next point that I would like to emphasize is that one that I said is the crowder, but then the DNA itself, that means the road condition, the road on which the protein moves, that has lots of unique features. So, for example, if you see the genomic DNA, major component of the DNA is that the first this DNA, if you stretch it, it will be around 2 meters long, the whole human genome. However, it fits within a very small space of a cell nucleus. So obvious way to do that is it undergoes a huge level of packing. So this packing is hierarchical in nature, and the fundamental unit of this packing is called nucleosome. So where this DNA wraps around this octameric histone protein roughly 1.67 times, and so which is around 150 base pairs, and then many of such nucleosomes come close to each other to form this chromatin fiber, and then higher order packing is there. But then the question is, we are trying to understand that as because these positions, the DNA is completely occupied by this histone octameric protein, then if there is a gene or there is a specific site on this nucleosomal DNA, how a protein can really read that site? So this is a closer and a clearer look of nucleosome, and this is within this octameric histone protein, and here we have around 150 base pair DNA. Fortunately, recent study suggests, published in Nature Structural Biology, it suggests that this nucleosome is not a simple, static packaging block for genomic DNA. Rather, it is a dynamic, has a dynamic nature, and can open from both the ends, like this kind of some set of DNA can peels off the histone surface, and therefore these positions are accessible for other proteins. So we wanted to see that this is called nucleosome breathing dynamics, and this happens with the rate k open and k close, let's say. So we tried to understand that how this breathing dynamics can influence the target search process of a protein if the sites are located somewhere here. So that means this whole segment can sometimes be wrapped, or sometimes it can be transiently open for the protein. So we were able to find out some kinetic rates of three proteins here. Out of these three proteins, these two proteins, they are known as pioneer factors, and LEX-A is a protein which is known as non-pioneer factor. Now, what is this pioneer and non-pioneer? Well, all these three proteins are basically transcription factors. Out of which these two proteins, which are pioneer factors, these proteins they have a specific nature that they can invade nucleosome very efficiently without help of anything. Whereas this non-pioneer factor, they cannot on their own invade nucleosome. Rather, they need help from pioneer factors. So we have this on rate and off rate of these proteins on nucleosomal DNA and on linear DNA, right? So these are the rates we have, and with that we are trying to build an analytical model. So what we have done here is that, let's say this is your nucleosomal DNA. So this green beads depicting your nucleosomal DNA, and this barrel type shape is basically the histone protein. So this red dot is the target site where the protein, this pioneer factor, needs to bind. So this is a pioneer factor. It can come with a rate k on and k off. So when with a rate k on, it can come and bind to some one place, and then as the nucleosome undergoes nucleosome breathing with a rate k open, as soon as it opens up, this pioneer factor can move one-dimensionally along the DNA. So when it is closed, the protein can come, but it cannot move one-dimensionally because these sites are otherwise occupied by the histone proteins. So it has to wait until this DNA segment opens up from the histone surface, and then it can perform this one-dimensional movement both the sides. And again, if it is wraps back, then again it stops, or the search process is halt. There is a halt, and it doesn't move along the DNA. So with this picture, we have tried to formulate the different phases. For example, this is you have two main phases. One is this state, which is we call fully wrapped state, FW, and this state we called PU, which is called partially unwrapped state. Now, let's say you have this PN state, fully wrapped state. What can happen? Okay, the fully wrapped state, it can go to the solution phase. From this state, it can go to the solution phase, the protein. or it can, with a rate k open, it can go to the partially unwrapped state. These are the two possibilities. This will happen to, you can see that 1 to length small l, right? The for the fully wrapped. Now, this region in the fully wrapped, you can see here the we assume that this segment, this is the small l segment, 1 to small l, this portion can unwrap. The rest of the segment will never unwrap from the DNA. So because in that case the nucleosome will completely the structure will be destroyed. So for the rest of the portion, there is no movement or the conformational change to the unwrapped state. So this is the for the L plus 1 to state L. Now, when it is in partially unwrapped state, for 1 to L, in addition to this, you can have now this one-dimensional movement on the DNA. Right, and for L plus 1 to L, this one-dimensional movement is not there. Otherwise, and also this the partially fully wrapped state, it is basically the same for that segment. And this is the solution phase. From solution phase, it can come on the DNA with this rate, and both the states are equally probable, so it can come on any state. So these two are the boundary conditions, and for this systems we can again do this the process we are already mentioned, that doing this Laplace transformation, and then we can find out this mean first passage time, which is given by this equation. So what we found here is that the time that we measured T0 on the nucleosome and T0 on linear DNA. Now, if you see these two, as I mentioned already, REB1 and CBF1, these two are pioneer factors, and this Lexe is non-pioneer factor. Now, on nucleosomal DNA, you can see the non-pioneer factor needs much more time to reach the target site, which is very, very high and unphysical. Secondly, this is the position of the target site where it is located from one terminal. So you can see as the target site is deeply buried inside the nucleosomal DNA, the time increases much more, right? It increases exponentially. But on the linear DNA, you can see that the non-pioneer factor, Lexe, is slightly faster, at least two-fold faster compared to the two pioneer factors. Okay, so we try to understand that what is so special with the pioneer factor that it invades the nucleosome so fast and it can reach their target site so efficiently, which is not there on the non-pioneer factors. So what we found here is that we plotted the target search time on the nucleosomal DNA with the rate k of what different k on. So what we found here is that there is a non-monotonic behavior of the time with the k of, and it suggests that there is a specific k of, intermediate k of is important for the fastest target search time. Now, more importantly is that if you see this different k on, as the k on increases, right? This is 0.0006, 0.001, and 0.01. As the k on increases, the minima shifts towards right. So that means with the change of k on, k off at which you get the fastest target search time is also shifting. So that means there is a correspondence between the target k on and k off to obtain the fastest target search time. So we have measured here the on rate and this resetting of the off rate. What we found here is that for the this red line shows you the fastest target search time, and both the pioneer factor and these two black lines are basically 10% deviation from this fastest target search time. And you can see that both these pioneer factors fall within this region, whereas this Lexe is much away from this region. It suggests that for the pioneer factors, there is a correspondence between the on rate and off rate. They should be quite comparable, which is not in the case of non-pioneer factors. So what happens that we hypothesize here is that for the pioneer factors, both the on rates and off rates are comparable. Now, if your on rate is very small, so that means probability to or the possibility to find out the DNA and staying on the DNA is very small. The residence time on the DNA is very small. So that means then your off rate should be very small as well. Otherwise, you will not, the protein will not get enough time to move on the DNA and find its target site. So very small on rate should be compensated by a very small off rate. Similarly, if your on rate is very high, then the protein is very sticky to the DNA, and then it can come and bind to any space, any site, and then it can also go in the wrong direction. For that, you need a off rate which can kick off the DNA from the DNA, kick off the protein DNA surface, and therefore this will balance or counter each other. So there should be a correspondence between these two rates. And here you have another nucleosome with which is partially unwrapped state, as you can see. Now this is basin 1 corresponding to this structure, this is basin 2 corresponding to this structure, and our motivation is to develop a two basin model so that we can study this interchange of the conformations very efficiently. So what we have done is we have developed a model where this delta is a parameter that modulates the relative stability between this basin 1 and basin 2. And we have a coupling constant gamma that smooths this energy surface, and it this coupling here, and this also denotes the barrier, the energy barrier between these two states. So basically, the ER1 is for the energy of this state R1, and ER2 for this. So for this kind of characteristic equation, this will give a non-trivial solution if this condition is met, from which you can find this double basin energy, which we did. And we have found a parameter chi, which kind of denotes the transition between these two states. So you can change this chi, you will see that this nucleosome undergoes many transitions between these two conformations. One is fully wrapped, one is partially unwrapped. So which is shown here in this movie as well. Okay, so with this, a double basin model of the system is ready with us, and now we wanted to study how proteins really move on this nucleosomal DNA. So we have two proteins, which is called which are known as SOX2 and OCT4. OCT4 is a protein with two domains, and this is a SOX2, and both these proteins are pioneer factors. Now we wanted to understand how they move on the DNA. So first, what we estimated is the diffusion coefficient of these two proteins individually. What we found is that OCT4 moves much faster, diffuses much faster on the DNA compared to SOX2. And the reason behind is that OCT4 is a protein, multimeric protein. So basically, it can, it's like it has two arms. One arm can it can stick to one part of the DNA, another arm it can use to stick another part of DNA, and then it can move from between these two sides. So it can move much more efficiently on the DNA compared to SOX2, which is a monomeric protein. So its diffusion is higher. Okay, fine. So diffusion is higher of OCT4. Now we wanted to understand whether the kinetics is along the line or not. So what we found is that we tried to find out the native contact formation probability, and for this case we found that SOX2 is faster compared to OCT4, if you see. So what is going on, right? So what we found is that if you analyze the trajectory and if you see the on rate and off rate from the nucleosomal DNA, we find that for SOX2, this the on rate and off rate, these are the results, which is much more comparable compared to off rates. If you see for SOX2, this is much more comparable on and off rate, but for OCT4, there is a huge discrepancy between these two. So as because this is much more compensated, the two rates, we understand that this SOX2 will move much faster on the DNA to recognize its target site. So its kinetics is faster. So this association compensated dissociation mechanism that we predicted from our analytical study holds true for this molecular dynamics study as well. Next, we try to understand what happens this when this both SOX2 and OCT4 is present. So there is a fantastic live cell study which suggests that SOX2 typically binds faster and then OCT4. And not only this, they also mentioned that this binding is cooperative. That means SOX2 binds and it will facilitate the binding of OCT4. So we try to understand whether we can capture that. If yes, then why this is so. So what we found is that if both the protein is present, then you can see SOX2 binds much faster compared to OCT4. And then to understand whether there is any cooperativity or not, we try to understand estimate the binding free energies. So we estimated the enthalpic and entropic contribution of these three systems individually: the free nucleosome, the nucleosome with SOX2 complex, and nucleosome with SOX2 and OCT4. What we found is that the net enthalpy change during the process is positive, which means enthalpically this is a forbidden case. However, if you see the entropic behavior, you can see that there is a large gain in entropy, and overall, this is entropically permitted or entropically favored process, that the binding of OCT4 is favored by the binding of SOX2. So we try to understand why this is so. What is the origin of this entropic, cooperativity? Sorry. So what we have done here is we have performed principal component analysis. So for that, we have estimated the positional covariance matrix, which is basically 3n by 3 matrix. And then we have diagonalized this matrix from the trajectory for each of this free nucleosomal DNA, nucleosomal SOX2 complex, and nucleosomal SOX2 OCT4 complex. So basically, the eigenvectors of this diagonal matrix is they provide a vectorial representation of each mode of structural deformation. And the eigenvalues represent the magnitude of this mode of structural deformation. So the highest eigenvalues denote the most significant contribution of corresponding mode. So here we have done this for these three systems, and for the three most important components, we have looked the behavior. This is the corresponding sequence. And what we find here is that you can see the major group with difference in these three cases, there is a string-like behavior as if there is a kind of wave moving on. So when it is a free nucleosomal DNA and when SOX2 binds, there is a huge change in this behavior. And you can see that, it becomes highly asymmetric and the higher harmonics predominate, which is again largely restored when OCT4 binds again with this, right? So this was kind of, it suggests that there is a dissipation of information from one side to another side, which is evident, but we wanted to get a deeper insight. So what we have done here is that we have estimated how the information is being transferred. So what we have done here, we have estimated the major groove with fluctuations, instantaneous major groove with fluctuations. This is the ensemble average. And then we have said that, okay, so this is the major groove with fluctuation, instantaneous, and then we can use this transfer entropy from the formulation of Schreiber, which is given by this. And then the conditional entropy in this case can be written as this equation, right? So once the conditional entropy is this, we can use this equation again here to find out the transfer entropy from one side to another side, I to j. And then the net entropy transfer between from residue I to residue j, we can find out by summing up the all contribution. So what we get with this calculation is that when there is no specific binding with the nucleosomal DNA, the SOX2 is not bound at all so far. This picture is similar like this. when SOX2 is bound, specific binding happens, then you can see there is an entropic funnel forms, right? So here there is a lower entropy, and here the entropy is quite higher. And if OCT4 is somewhere located in this region, it can eventually sense this, the gradient of this entropy, and it can move towards that direction. And when it binds, again, it restores back close to the average, close to the zero, which gives an idea that it happens somewhere like this, that because of this breathing, this is the binding sites of SOX2 and OCT4. What happens is that when SOX2 binds, it creates an entropy sink here. And then if the second transcription factor, which is OCT4, if it is located somewhere here, it can move towards this direction and can bind here. And once it is once it is bound, then this entropy, this information transfer stops, and then it completes the process. So this entropy transfer is basically a driving force here, which is the source of cooperative binding of this OCT4 following the binding of SOX2. We recently found that this entropy transfer mechanism is a generic mechanism for many cases. For example, you have a DNA and you have a mismatch. It happens very often on DNA, and there are a class of protein called mismatch repair protein, which comes and fix this problem. So what happens when this mismatch is there, that typically we know A, T, these two are the DNA base pairs that forms hydrogen bond, and GC forms a pair. But here, let's say A and A or T and T. So obviously they cannot form this hydrogen bond. So what happens is base can, this is much more free compared to other bases. And then it can, show this kind of flipping mechanism. So this flipping, what happens? So you see that if you perform an atomistic simulation and we measure the distance of these base pairs, so we see that the distribution of this, base pair distance can be correctly predicted using our coarse-grained model as well. So what is our motivation is to try to understand that, compared to a normal transcription factor binding, DNA mismatch repair involves only one single base, and how proteins can find this single base out of many DNA base pairs, right? So to understand this process, we first tested whether our coarse-grained model can capture this kind of distribution predicted by the atomistic simulation or not. And once that is done, we proceed towards understanding again the search process. We again found that the net entropy transfer, when you have a mismatch, you have a funnel-like structure. When you don't have any mismatch, there is no funnel-like structure. And not only this, what we found is that this net entropy transfer, if you consider at different times, and if you find out the autocorrelation of this entropy transfer, you will see a periodicity in this transfer. That means there is a signal that is coming out periodically from this site of mismatch because of this base flipping, which is not present in the case where DNA mismatch is not there. And our argument is that the protein can, if it is nearby, it can sense this periodic signal and can move towards that direction. So interestingly, if you see the dynamics of the protein, the sliding, hopping, and diffusion, when mismatch is present and there is no mismatch, there is not much difference. So basically, the dynamics is not different. However, the kinetics is different. If you can see here, mismatch is present, the protein can more efficiently go towards there and can bind towards to the site. So again, we found that the net entropy transfer, when there is a mismatch, you have a funnel, and this funnel is around 15-20 base pairs from far away the protein can sense. And once it binds, there is again the restoring the whole entropy transfer mechanism. And this is again, as I said, that one possible mechanism, one generic mechanism that explains many target search process, many the reason behind facilitated diffusion of proteins on DNA. The last point that I would like to cover here is that the road is, as I said, there is many unique features on the road, that the DNA. So I discussed on this portion, which was nucleosomes. If you consider much hierarchical organization, then you can see that the chromosome organized into different compartments, which is known as A and B compartments, or some compartments are transcriptionally active, some compartments are not. So it was found that these transcriptionally active compartments, they are typically uh, negatively supercoiled DNA, and where the compartments are transcriptionally inactive, they are typically positively supercoiled DNA. So this supercoiling topology is kind of also important, possibly because of to regulate the transcription process. So to understand this, we want to create another model and to try to see whether we can predict this, supercoiling topology and how that regulates the search process. So here we have a DNA mini circle, and we have developed a DNA model that can accurately predict this, supercoiling. And we have compared our supercoil structures with many cryo-EM structures, and we found quite a decent match. Now, with this, we see that the DNA can predict these supercoiling structures. So we try to understand how protein moves on the supercoiled DNA. So we have again measured the diffusivity of the protein on the DNA with different linking number. So this linking number denotes that how much supercoiled the structure is. And as you can see, if the supercoiling is more, the DNA can assume a plectronomic structure, which is kind of this kind of twisted structure like 8, and or it can be more such structures can happen more in this structure. For example, two DNA segments come close to each other only once. It can be more than that. So we found this diffusivity, and what we found, the genetic feature is that on negatively supercoiled DNA, you can see the diffusivity is much higher compared to positively supercoiled DNA. And to understand why it is so, what we found is that if you consider, so what happens is, see, when you have a negatively supercoiled DNA, the stacking of the bases is destroyed, right? So what happens is that as the stacking is destroyed, the DNA wants to restore its stacking, and therefore, if there is a because of this supercoiling, if there is a change in the stacking, it wants to send the signals further so that it can restore the stacking here, and then this movement goes on along the DNA. So there is a torsional stress that proceeds along the DNA, and we wanted to measure this torsional stress. So what we have done here is that we calculate this torsional, the movement of this torsional stress. So for that, we have calculated this autocorrelation of this factor of this vector Ci. So Ci is a vector that is basically can be found out from the cross product of Ai and Bi two vectors, where Ai defines the unit vectors connecting the midpoints of ith base pair and the I plus 1th base pair, and Bi represents the binormal vector. So we wanted to see the autocorrelation of this Ci vector. And what we found is that this kind of scenario can be observed for a linking negatively supercoiled DNA and a positively supercoiled DNA. Now, this Ci value, if it is close to 1, it suggests that the DNA segments that are yet to undergo twisting. Whereas if Ci is very less than 1, it indicates the segments has already undergone twisting. So if you see the negatively supercoiled DNA, you can see there is a slope here, and that means there is a the twist has moved from one position to another position on negatively supercoiled DNA within this lag time, whereas on positively supercoiled DNA that hasn't happened. That suggests what happens is that these two positions where the DNA come close to each other kind of intersect. These two positions can move quite variedly along the time with the time along this DNA contour. Whereas in positively supercoiled DNA, it is almost fixed. And what is the impact of that? The impact is that a protein which comes at this position can again do intersegmental transfer that we already have mentioned previously from one side to another side, and therefore on the negatively supercoiled DNA, the protein can scan many positions on the DNA whereas on positively supercoiled DNA, it will not be able to do so. And that gives an added advantage to the proteins on negatively supercoiled DNA to communicate between many DNA sites very efficiently. So in this plot, you can see that most of the DNA has been scanned by the protein when it is negatively supercoiled, whereas for positively supercoiled, it isn't. And it is, and if you see this red color is basically the residency time. So this residence time, you can see it is quite red here. That means the protein remains mostly at this region for very long time, and it could not move much faster. Whereas because of this twist propagation along the DNA or negatively supercoiled topology, the protein could move much faster and scan the DNA much efficiently. So this explains our study explains why the transcriptionally active sites are preferably adapt negatively supercoiled topology. We also showed that the radius of gyration of these supercoiled minicircles is that when you have negatively supercoiled structure, you don't see a much change with presence and in absence of the protein. Whereas the positively supercoiled topology DNA, in the presence of protein, it undergoes a compaction. And that also is in line with the experimental observations that the transcriptionally inactive domains of genomic DNA that adapts positively supercoiled topology are much more compact, and therefore the sites that are of interest, DNA sites that are of interest, are occasionally not accessible by the proteins. So in conclusion, we would like to mention here that typically this search process happens by two ways. One is called DNA-based readout. Based readout means that the protein comes at the target site, and then it forms specific interactions, which are mainly hydrogen bond interactions with the bases. But as you can see, these are very specific interactions, and this hydrogen bond interaction, it needs very close proximity between this protein and the specific DNA site. So this happens only when the protein is very close to each other, close to the DNA. Another mechanism is called DNA shape readout, where the protein can sense the changes in the DNA shape that can be caused by the DNA sequence or can be caused by binding of other protein. And here we argued that this change of shape can actually generate a signal which moves along the DNA contour, and a protein from a far distance can sense that signal and can move towards this binding site very efficiently. So we kind of give an alternative, a generic mechanism of DNA shape readout. We also suggest that nuclear crowding modulates this target search efficiency of the DNA binding protein. And nucleosome dynamics is important to access the DNA sites that are otherwise occluded. And last but not the least, the DNA topology, the supercoiled topology we are referring here. The DNA topology regulates the protein communications on DNA, and typically negatively supercoiled topology is much more preferred for DNA, genomically, transcriptionally active genomic DNA, right? So these are the major conclusions, and I hope I could show you that some of the important rules that govern these search mechanisms of protein on DNA. So the work is mainly done by Anupam, Shujit, and Sangeeta, and another student by Pinky, who has done the Crowder's work. And Anupam and Pinky both are doing postdocs now, and these two are almost about to finish. And I would like to thank my funding agencies, and would be happy to answer questions if there are any.
Yeah, thank you, Arnab. Really fascinating talk. Lots of interesting results. So now the floor is open for questions. Please raise your virtual hand if you want to ask the question yourself. If you have any question, please raise your hand. Meanwhile, there was a question in the chat box from Ravi. But Ravi, if you are around and if you would like to ask the question yourself, you may do so.
Yeah. Hi, Arnab. Very nice talk. A lot of interesting results, as Suman mentioned. So this question is, like you mentioned about this macromolecules or the crowding molecules not entering the depletion region. So are the reasons known? Why is it so?
As I said, this depletion region, this is a quite known phenomena when you have molecules of different sizes. There is
Okay.
a entropic, entropic origin of this because of this, disparity in their sizes. This
Okay.
depletion happens or creates within which the crowded molecules cannot enter. So
So
this
why?
is kind of because of mainly, obviously, as I said, the one can assume that this is volume interaction, but this isn't, because even if you do not have, if you switch off the excluded volume interactions, because of the entropy of these two different molecules, there is a space that remains intact, and another molecules cannot enter within this region. So the nature, the reason, underlying reason is purely entropic.
So if I use a crowding molecule of one type or one size, okay.
Yeah, then it is solely governed by the excluded volume interactions.
Okay. Okay. One more question, maybe can I ask?
Yeah, please go
Yeah,
ahead.
sure.
So this you mentioned about this k on of values, right? K
Right.
on in one of, I think probably slide 15 or 16 on that. So they are like
Yeah.
orders of magnitude different. So
Right.
are these your assumed values or they're known experimentally?
No, these
Such
are
big,
all experimental values. That is
so
what I'm
such
saying.
big,
Sorry, yes, please go on.
no, can you go back to the slide, and then such a huge variation? Why is it so?
Yeah, just a second. Allow me. Uh, I don't know, it has stopped maybe. Let's see again.
Yeah, you can try
Just
sharing again.
uh, it's here. Is it visible?
Yeah.
Yeah.
Yeah. So yeah. So if you see nucleosomal case, the on rates are very much similar. If you
Okay.
see on the nucleosomal 10 to the power minus 4, 10 to the power minus 4, 10 to the power minus 5, right? For all the three proteins. However, if you see the off rate, huge discrepancy.
Okay.
And this is the that is what we are arguing, that this is the kinetic origin why these two proteins are can have pioneering activity. It can invade the nucleosome, and why this protein is not able to invade nucleosome on its own, because of its high dissociation rate, right? Because as the nucleosomal DNA does this kind of dynamics. So the protein should have a high affinity towards the DNA. Otherwise, what will happen every time it goes off from the DNA, then it will not get enough time to move along the DNA and search for its side. So this high dissociation rates is a kind of culprit for this kind of behavior for non-pioneer activity.
So is it
So yeah.
is it known why this is it related to the shape or size, or means like are that known?
Well, there is no such generalization why these on rates and off rates and how they are dependent on the structure or sequence of the proteins. But it is true that there are certain proteins known which have pioneering activity, and there are many transcription factors which have no pioneering activity.
Okay, so that's
So we
interesting.
found the on and off rates of these pioneer proteins. Fortunately for these three proteins, there are not much information about this, but definitely it is an interesting question to understand why or what structural or sequence property makes this kind of on and off rate on DNA.
Can I just ask one simple basic question? Maybe I'm not working in this, so I cannot, don't know. So what is the origin of this transfer entropy? Is there a like driving force, or what is the driving force, or what is the origin of this?
So, right. So basically, the idea is that DNA has its plasticity, right? The DNA tries to maintain its structure, and when there is a deformation or when there is a change in the local geometry,
Maybe there is some network issue.
Network issue.
Okay, meanwhile, I'll try to call him and check. Yeah.
Okay.
Okay, Arnab, there was some network problem, I guess. You are muted. You are on mute.
right? I don't know how far I could hear, but what I was saying is that yeah, there is a if there is a structural deformation, the DNA tries to regain back its own structure. And while doing so, it tries to dissipate that deformation from one point to the near and adjacent sites. And that information that information transfers from one point to another, which is the origin of this entropy transfer mechanism.
So can this affect your K of value? Can your K of values get affected by this sort of dissipation transfer?
I guess not very much,
Okay.
because K of is mostly the property of the protein, and the kind of this entropy transfer mechanism I have shown, this happens around the segment of the DNA is around 30 to 40 base pairs.
Sure.
So typically protein, the scanning length of protein, that means the length of the DNA that protein scans without dissociating from the DNA is typically in this order. So protein can definitely dissociate from this segment as well. But as what I was emphasizing here is that the search process, the search of the protein is more directed towards a direction in this region of the DNA. For other region, there is no such directed search. Otherwise, the K of should not be altered too much.
Thank you. Thank you very much.
Thank you. So let's go to the next question. Shushmita, please go ahead.
Very interesting talk. Actually, I was thinking about when you say that when you actually modulated one base of the DNA flipped out, and then you were was quite evidently you got the entropic funnel. So my question is that are you specifying the protein, means the protein here to be uh, associated with DNA specifically or non-specifically? And second question is that the binding site is the flip-flop site or is quite distant?
Okay, the first question is when we try to see this entropic funnel, we did it only with the DNA with one site mismatch. So that means there is no role of the protein in creating this funnel, number one.
Okay.
Although we have seen that when the protein is present, still this funnel is there, but when the protein binds at this site, then the funnel disappears. So this answers your first question, that it is from the flip-flop mechanism. And secondly, the binding site is obviously the lesson or the site at which there is a mismatch, so that the protein can come and repair this mismatch. So exactly the same site.
I see. I see. Okay, thank you for that. And I have another question, which is related to your first introduction, where you were saying that when protein moves quite faster, then uh, there is a chance of not trapping into the potholes. So, uh, is there any experimental evidence known for that? Means for at least for this protein DNA system, that when it speeds up for roads, of course we know that when there is less surface interaction, then but for specifically in this kind of biological system, is there any known
Yes.
experiment?
Yes, there are some experiments. So basically, in reality, what happens if you see the DNA, this specific site, if we call them genes, there
Mhm.
are some other sites which are called as enhancers,
Hmm.
right? So the role of these enhancer sites is basically they are like antenna. So let's
Hmm.
say you have this is your specific site and this is your antenna. So the antenna, this site has quite sequence similarity with this target site.
Hmm.
So the role of this antenna is that if the protein is nearby, the protein will see this antenna, which are pseudocytes, and then it will be close to the DNA. It will not try to dissociate anymore, right?
Hmm.
But if this enhancer is far away,
Hmm.
then the impact of this enhancer is no more. Then the protein can sense this, but while traveling in between, the effect
Hmm.
of antenna is not there anymore, enhancer is not there
Hmm.
anymore, and then the protein can dissociate back. So the role of
Hmm.
this enhancer is to compel the protein to look the DNA bases very carefully so that it
Hmm.
doesn't go away. Because if
Hmm.
it goes away, it will not read the correct DNA bases properly. So there are experiments which looked into this role of these enhancers, their positions, and how do they impact the search.
No, my explicit question was like kinetically, the speeding up process means diffusion, the variation of the diffusion and then the search. Is there any known experiment, not like with
Yes.
the in the presence of enhancer or a with the
No,
presence of
yeah, that is what I'm saying. There
hmm,
are experiments you can see by there are several NMR experiments by I forgot name. Maybe I can give you the
okay,
details
I
over
can
email.
then yeah, okay, I
Sure.
can communicate with you through
There are
email.
experiments.
Okay,
Yes.
all right, okay, thank you, Arnab.
Thank you.
Thank you, Shushmita. Anybody else? Any other question? Okay, I had a couple of kind of naive questions. So regarding your first part, so where you are talking about protein moving along the DNA. So does it require? I mean, does it involve any displacement of water? Because we know that there is a spine of water along the DNA, right? And now when the protein binds, does it need to displace some of those bound water? I mean, because that happens when, let's say, protein ligand binding happens. Often some bound water gets released. Is there any such phenomena involved here?
Right. This is well, two answers to this. One is that first of all, our model is a coarse-grained model,
Yeah, I
so we
understand.
don't have any water molecules.
Mhm.
However, we can definitely comment on this. The comment is that the Crowder molecules I have shown, and we already, as I mentioned, that in presence of the Crowder molecules, you get a depletion layer, right? Depletion region. So the one can measure the width of this depletion region, and if you measure the width of this depletion region, it is very similar to the third hydration shell of water around DNA. And a recent work by Shobhan's group they have done, and where they have also shown that when the ligand is within this depletion region, process this hydration shell, the Crowder impact is not anymore experienced on the molecule. But in the bulk, definitely there is. So yes, the hydration layer is there, and it when the if the protein is big in size, definitely it can distort that surrounding water molecule. But if it is small one, it may it may be much closer to the DNA, and the hydrogen second and third hydrogen layer will remain probably intact. The first hydrogen layer will get distorted. That is my assumption.
Right, right. Thank you. And the next question was, I was kind of curious to know about this nucleosome. So wrapping around the nucleosome, and you
Right.
mentioned the loosening of those. Now, I was wondering that since it's a long kind of polymer chain, so when it has to loosen up these knots, the contour length has to increase, right? But so there will be some constraint from the like end-to-end distance. Is that a factor or not?
No. No, this is you have to see that this is the histone code, and the DNA is wrapped around it. So there is no loosening up, because loosening up means you are kind of elimination of all the histone DNA contacts. So simultaneously that will not happen, because that is free energetically very highly costly. So what happens is from one end of the histone, this nucleosome, the DNA partially unwraps. So that is of much lower cost energetically. So one segment can unwraps and
Only at
rewraps
the
back.
end, in one end
Yeah,
only.
towards the end. Yes, yes. But there are several other molecules which are called nucleosome remodeler. So this can come and bind to even unwrap even half of the nucleosomes. That happens.
But what happens during the very first step of packing? I'm just curious that, so let's say when it has to pack to begin with, I don't know what is the
Right.
T equals to 0. So let's say
Yeah.
I have a kind of a disordered chain. Now it has to wrap. So now it's a very
Right.
difficult process, right? So how does it happen?
So typically, if you see the nucleosome structure, there are, as I said, almost 150 base pair wraps around this octameric histone, and there are typically 14 sites, 14 minor groove widths, where this DNA comes very close to the histone protein, and there is almost specific interaction between them. This position at these positions. So I suspect that this happens sequentially, and when it tries to wrap around the nucleosome, it goes sequentially, the DNA moves around it, and this sequential
Again, it has to happen through one end, because if it has to wrap in the middle, then that's what I was thinking, that the whole chain needs to move, right? If it has to happen
right,
in the middle,
right,
it can
right,
happen only in a directional manner in one end, from one end.
right, right. In fact, there are evidences where it is shown that let's say you have a chain, and here one histone protein is there, which is forming the nucleosome, that can move along the chain, which is called nucleosome sliding.
I see.
So that means this the position of nucleosome can change, and therefore, the site accessibility of these different DNA sites will vary with time, because this with the nucleosome repositioning, different sites will be blocked or open. And when open, there will be protein can be transcriptionally, the transcription can happen, and when blocked, transcription will not happen. So this accessibility is largely governed by this the packing of this nucleosome and hierarchical packing and all.
Okay. Very interesting.
This is quite multi-layered, actually.
Yeah, thank you. Yeah.
Yep.
Any other question from anybody else? Okay. If not, okay. Ravi has another question. Yeah. Yeah. Please go ahead. Yeah. Maybe one last question. Yeah.
Yeah. This is you mentioned about so your coarse grain modeling. Generally, this DNA binding proteins, there's mostly the interactions are electrostatic, right? So your coarse grain model, does it also include the electrostatic component?
Yes, it is mainly driven by electrostatics, as I said. So we have considered electrostatics.
And other, so you mentioned about that single base, like you can identify with coarse grain. So it can just give for throw some light on. That's very interesting.
Yeah. So again, what I mentioned before is that the single site mismatch, basically what happens when it is quite well-known phenomena that the base where there is a mismatch, it can do this kind of flipping. And when it flips back and forth, it creates a signal from this side. So there is an entropic signal again, because it is deforming the local geometry of the DNA, local groove width. And while it is doing so, that signal is dissipated from this side along both the direction along the DNA. And that can be sensed if the protein is nearby, around 20-30 base pairs away. It can sense and it can move along this direction.
So what you're saying
In fact,
is that
recently, I'll tell you, recently one JAX paper is published 2022 by Peter Olines, and where they have shown that there is a change in electrostatic surface, electrostatic potential surface, because of this, not flip-flop. They mentioned their DNA shape readout, not the single base pair. But they said that there is a change in this kind of electrostatic surface, potential surface, and the width they have shown, from which the protein can sense that probably the target site is ahead. That is around again 15 to 20 base pairs, which is very much consistent with our result.
But that costs energy, right? So where is the energy coming from? Or the driving
That
force?
for what?
For the
Flip-flop?
Yeah.
Yeah, because obviously when you don't have any mismatch, there is a hydrogen bond between these two, right? ATGC.
So it has.
So this hydrogen bond is there. But when there is no hydrogen bond, it is very free. It is very free. It can move like this and this.
So what you're saying is that the electrostatics of that area changes so that there is greater protein.
The electrostatic surface is again, I have shown in one of my nucleic acid research paper back in 2014 that this electrostatic surface of DNA is very much coupled with its groove geometry. So if the groove geometry changes, immediately its electrostatic potential surface changes.
So that brings in the repair molecules.
Right. So, but my point here is that when this flip-flop mechanism is there, it creates a signal which is there, and there is a periodicity of this signal, and this signal transfers from one point to another point, which can be sensed by the protein.
So your coarse grain model, like what is the one bead? How many bases are you including?
For
In your coarse grain model, one bead, how many bases?
One bead. So basically, every nucleotide is represented by three sites, three beads. So one sugar, one phosphate, and one nucleoside base. So basically, we have a very detailed description of the backbone of the DNA, and the structure, the resolution is so that you can easily transfer it back to atomistic representation.
Very nice, very nice. Thank you.
Thanks.
Okay, so if there are no further questions, on behalf of my other co-organizers, Professor Ranjit Biswas, Dr. Rajiv Biswas, and myself, I thank Dr. Arnab Bhattacherjee once again for accepting our invitation and delivering this excellent talk. I also thank all the participants who are here today. So yeah, thanks Arnab once again.
Thank you. Thank you very much.
And for others, we are going to announce the next talk very soon. If you have not yet subscribed to our mailing list, if you do so, you will get to know the announcement of the next lecture, hopefully sometime in early April next month. And yeah, so till then, goodbye everybody and take care. Stay safe. Okay.
Bye.
Bye. Bye everybody.